Okay, we're going to go ahead and get started. It is 4 o'clock on my clock, and we've got our guest speaker and our panel assembled. So I want to go ahead and welcome everybody to this very special webinar. This is entitled the COVID-19 Vaccine Research Updates and Recent Experiences. And this is really the first time that TRI has done something of this magnitude, and we have over 450 folks that have signed up for this webinar. So this is, of course, illustrates the great interest across our institution in the topic. I wanted to provide just a few housekeeping rules as we start into this. I'm Dr. Laura James. I'm the director of our Translational Research Institute here, which is our CTSA grant, and just wanted to provide a little bit of structure for how things will go this afternoon, and then I'll introduce our guest speaker, Dr. Pat Winokur. Dr. Winokur, as you're going to see very quickly, is an expert in COVID-19 vaccines as well as a lot of other vaccine-related issues. She's going to speak for about 30 minutes this afternoon. And then we're going to open the session up for a Q&A period. As we move into the Q&A period, we're going to switch the format to a panel discussion, and I'm going to go ahead and introduce those individuals, those that are participating in our panel this afternoon. In addition to Dr. Winokur, we have Dr. Bob Hopkins, who is chair of the National Vaccine Advisory Committee. He's one of our own. He's a professor of internal medicine and pediatrics. And then we have Pat Winokur. Thirdly, we have Dr. Chris Patterson. She's an associate professor here in the College of Medicine in the Division of Infectious Disease. And finally, we have Dr. Brian Gittens, who is our Vice Chancellor for Diversity, Equity, and Inclusion. So those four individuals will constitute our panel. I know that as Dr. Winokur begins to speak, a lot of questions are going to come forward. I'm going to ask individuals, as you have questions, if you will send those directly via chat to Dr. Bob Hopkins, and he will be the one fielding and refereeing the questions and pulling the questions forward as we move into that session of our webinar this afternoon. So it gives me great pleasure to introduce to the UAMS community Dr. Pat Winokur. One of my privileges in serving as the PI for the CTSA is really getting to meet some national experts across the country, and I definitely have enjoyed getting to know Dr. Winokur over the last 18 months. She serves as the Executive Dean of the Carver College of Medicine at the University of Iowa. She's a professor of internal medicine and obviously an expert in infectious diseases. She leads the CTSA program at the University of Iowa, and she leads the vaccine and treatment evaluation unit for that institution. So she is an incredibly busy lady, and amazingly, anytime I email her, she always emails me back. So thank you, Pat. I really appreciate your support in answering my questions that have come up over the last several months. Dr. Winokur is a graduate of the Washington University College of Medicine. She completed her internship and residency at the University of Iowa, and then went on to the NIH for postdoctoral fellowship training. Prior to becoming a COVID-19 expert, she has worked across the range of research, beginning with basic and going forward with translational and clinical research. She has expertise across a large number of areas. She started working in human papillomavirus, moved into antibiotic resistance, bioterrorism, influenza vaccine research, just to name a few. And when looking around this afternoon for some special tidbits about Pat, one of the things that really struck me was that I knew that Pat had led several COVID-19 vaccine studies, but she also enrolled 270 diverse individuals in the Pfizer vaccine study, and I think that's very relevant to our institution and so much about of what we're interested in today. So without any further ado, I want to welcome Dr. Winokur and pass the floor over to her. Thank you, Pat.
So I'm going to just try and go through a brief snapshot. This is a topic that is changing hour by hour, and I make my slides, as I said, in the morning, and I change them by the afternoon, but it'll give you some background on what you're seeing. And Laura asked me to talk about some of the science behind some of the reactions that you're seeing in the vaccines and some of the advantages of the different platforms. So I think the title, Operation Warp Speed, has been a blessing and a curse for most of us. The blessing is that things are happening at warp speed, and there have been some amazing administrative decisions that have been made that have really brought vaccines to us very quickly. I think it has increased the consternation of the public that they think that we have moved too fast. But there are a couple things I want you to think about. If you really look at how we've typically developed vaccines, I think the Ebola bar down at the bottom is a good one to look at. In 1976, we identified the Ebola virus. We spent 27 years looking at the test tube, trying to figure out what this virus was, what the proteins were that were most immunogenic. And then after 27 years of playing in the test tube and maybe some animals, we started making vaccines. And this vaccine was approved for the first time last year, 2019. Now skip up to COVID-19, and you've got this really compressed time frame. But a lot of the compression is because there's been tremendous science that was done with SARS coronavirus that started in 2002, and then MERS coronavirus that was identified in around 2012. There were a lot of investigators, not a lot, actually a small number of investigators that hopped on these two previous coronaviruses to understand the science. What parts of the virus were immunogenic? They started developing vaccines that were tested in animals. And when COVID-19 came around and we found that it was a coronavirus that had a lot of similarities to SARS and MERS, that really jump-started the science pretty dramatically. So we were very lucky that we had that previous investment in science by some very dedicated scientists. If you look at what they had found with SARS and MERS, they had identified the spike protein as being the most important piece of the coronavirus. This was the protein that binds to the ACE2 receptors that are scattered in a lot of cells around the body. They were able to slice and dice the spike protein. They found in some studies that if you really honed in on the receptor binding domain, which is the copper color, that they got more immunogenicity than the full spike. Others found the full spike was better. They were able to actually mutate a couple prolines that are in the center of this receptor binding domain. What those mutations did is they really locked the construct into the pre-fusion form of the spike, which is the form that you would want to develop an antibody to prevent it from binding to the cell receptors. And so a lot of work had been done. There was one finding that they found when they put these vaccines into animals that was something that has come back to be an important focus for the studies that we'll be talking about later. And that was in some of the vaccines in animal models, they showed that the vaccines actually enhanced respiratory disease once the animals were then challenged with the coronavirus. So that's an important thing to think about. There have been quite a number of vaccines that have made it into human studies. There are 13 now that are in phase 3 studies around the world. A lot of the fastest ones are more genetically based. Initially, people have played with DNA vaccines And really remember, another thing that's helped us is that for the past 15 to 20 years, we've been trying to develop enhanced vaccine platforms. When you look at the DNA vaccines, one of the things that people have worried a little bit about is that the DNA does get into the nucleus, and there is the remote possibility that there could be genetic insertions. The mRNA vaccines have really kind of allowed us to bypass that concern, because the mRNA, as you know, is an unstable molecule and gets broken down quite quickly, and we don't typically integrate RNA into our DNA. And then there's been a lot of work going on over time with a variety of different viral vector platforms that really use the carrier of the virus to bring in the DNA or RNA, the genetic code for the spike protein. So when you look at the mRNA and viral vectors, there's some pros and cons to each that we'll go through. So when we started looking at the concept of having mRNA platforms, one of the difficulties is that there are no licensed mRNA vaccines. And for those of us that are in the vaccine world, that's a little bit of a concern, because, you know, how willing is the FDA or the US public going to be to accept a new platform that we have no previously licensed vaccines? They are fast to create and produce, as we have seen. They are highly unstable. mRNA is an unstable molecule, but they created ways that they could coat the mRNA in lipid nanoparticles that really helped protect the RNA before it got into the cell. They do still need ultra cold storage, which is not a great thing when you're thinking about vaccines. These platforms do stimulate the pathogen-associated molecular pattern receptors, which is part of the innate immune activation, and I will talk about that in a moment. But they've been able to really work with these mRNA platforms to modify the 5' and 3' untranslated regions. They were able to use strategies to replace kind of rare nucleotides with more commonly found tRNA species. All of these were able to enhance the protein translation of these mRNAs to create the proteins. Some of the platforms, not the ones that are moving into phase 3 or into FDA approval, have the ability to self-replicate, but that's something that we may be seeing in the phase 2 of the mRNA vaccines. If you look at the viral vectors, there is one licensed adeno-26 Ebola vaccine, and that was interesting because it starts with an ad26 prime, and then you're boosted with a vaccinia virus that's expressing the same protein. There are also VSV proteins or vaccines that have been established for Ebola. They can be replicating or non-replicating. Again, they're fast to create and produce. They're more stable in mRNA, usually stable at -20, which is a much improved stability condition for distribution. The problem with the adenoviruses is that if you have pre-existing immunity, it does limit the immune response. And they have been playing with the different adenovectors. In different countries, we have different strains of adenoviruses that are circulating. Africa, in particular, has an ability to use adeno5, but that wouldn't be a good choice in the US. And so there are different strategies for different countries that might be more appropriate. They too can stimulate these pathogen-associated molecular patterns and stimulate an innate immune response. But because of that pre-existing immunity issue, there is the possibility that this may be a kind of one and done phenomenon, which is we may use it for COVID-19, but maybe it won't be great for other vaccines in the future. That's something we're going to have to learn. If you look at the mRNA vaccines, once they're coated in this lipid nanoparticle, they're brought into the cell. where they're translated by your own cellular ribosomes. One of the nice things about that is when they create the spike protein, they're creating it very naturally in the same way that your cell would create the spike protein in a natural infection. And so then it is exposed on the surface, and the immune cells can be recognizing it and creating the B and T cell responses. The adenoviruses are somewhat similar. It's just they're actually using the adenovirus capsid to bind to the cell. They do have to get into the cell nucleus, where they are then translated, transcribed and translated into proteins, again expressed on the surface. The interesting thing is both of these can trigger this activation of the innate immune system. And that is an important thing to think about, because as you probably remember from Immunology 101, or don't remember from Immunology 101, is these pathogen-associated molecular patterns are designed to recognize small molecular motifs by the toll-like receptors. And these are things like LPS on gram-negative bacteria. They recognize double-stranded RNA because that's not a natural substance in the human body. They recognize some of these nucleic acid variants like unmethylated CpG. Those are all kind of designed to recognize foreign invaders. And the innate immune system then swoops in and creates an immune response that helps you kind of grab and phagocytose and kill these invaders before you have the 10 days to 2 week time frame it takes to start to develop an immune response. They do trigger a signaling cascade that stimulates interferons and other cytokines. And so that has good things and bad things, because the innate immune system is a nice way of creating an adjuvant type activity for these vaccines, but it also causes a lot of reactogenicity, fevers, other things like that. And so when you're developing these mRNA or other vectors, you try and balance that adjuvant behavior with the reactogenicity. And they found that they really did need to try and modify these to reduce that innate immune response to make them more palatable. A lot of the times they were able to modify some of these unusual nucleosides that the body picks up as being foreign, and substituted different nucleosides that allowed kind of a dampening down of that triggering of the toll-like receptors. The other thing that's happened with these vaccines is they've had to undergo really intense purification, because you do not want any of your vaccine to have the double-stranded RNA. And there are ways of purifying that out in the manufacturing protocols. So that's been a way that they've been able to modify these vaccine platforms to make them more effective and more tolerated. Most of these phase 3 trials that we're starting to see results from are designed pretty similarly. Most are randomizing 1-to-1. That gives you a little bit more statistical power than, say, a 2-to-1 randomization. Most of them have 30,000 participants or more. Most of the vaccines are requiring 2 doses, and that's not uncommon with these new pathogens that you have to prime and get the immune system kind of ready to boost with a second dose and get the highest immune response. FDA has issued guidance for the companies. This came out in October of this year, and they said that they would consider emergency use approval if these vaccines showed an efficacy of 50%, which means that they have a 50% chance of reducing symptomatic COVID-19 infections. And it typically is set at a week to 2 weeks after the second dose of vaccine. Safety, we always are looking at safety in vaccines, and that's no different with an emergency use authorization versus full FDA approval. we always are focusing on adverse events, especially these rare events like new immune-mediated diseases, Guillain-Barré, transverse myelitis. We're also looking for cases of severe COVID-19, because remember, I told you in SARS and MERS, some of the animal studies had shown that vaccines in those pathogens could actually enhance the severity of disease. So FDA said you have to have a certain number of severe diseases, and they said 6, to be able to show that they're happening more in the placebo group than the vaccine group. They said follow-up would have to be at least 2 months after at least half the people had reached their second dose. And again, enough cases of severe disease to ensure that, or help ensure, and remember statistics, when you have rare events, statistics have broad confidence intervals, but it would help them be confident that they're not seeing enhanced disease. So the big news, and many of you may be dissecting this data at the same time that I have been, FDA has been very transparent with this data, and it's much appreciated for all of us that are looking at the data and trying not to take all of our information from the New York Times or other publications. This came out on the 7th or 8th, and it is the FDA analysis of Pfizer's data. So this is actually a second review by experts, not just from the company. And you can see in this data that the Pfizer data, which is the first to go to the emergency use evaluation, had about a 95% efficacy, and that's in all groups. What we saw for the first time in this data set, or the first time I've seen it, was we started to be able to see some of the subsets. And you can see here, one of the subsets we're always most worried about are the 65 and older crowd, because that's a group that has immunosenescence, and you're hoping that they have an equal efficacy. And you can see here that the 65 to 74-year-olds, 93% efficacy, which is exceptionally good. The 75 and older, not a ton of cases, but it was 100% efficacious in the small numbers of cases they saw. That is fantastic news. And they were able to look at those who had risk factors for severe disease. Again, 94 to 95% efficacy, remarkably successful. They showed the data for Black Americans. They showed the data for Hispanics, those who are obese. They broke it down as best they could, and the efficacy each time was exceptionally good and very similar to this 95%. This is the incidence of COVID-19 infections as people started to accumulate data. And this starts from the day of first vaccination at day 0. The red line is the placebo group, and the blue is the vaccinated group. And you can see you start to diverge between the placebo group and the vaccine group, even after 10 days. And one of the things that they were able to show is that even between dose 1 and dose 2, the efficacy was not as good. It was about 52%, but it was still better than nothing. And so that was another piece that was great to see. This is from the phase 1 study that was published in New England Journal in October. And this is the immune response by antibody titers. These are neutralizing titers. So this is the ability to actually truncate productive infection in human serum. And this is I pulled out bits and pieces of this to show you just the product that went into phase 3. And you can see here that day 28 is 7 days after the second dose of vaccine. There's a really nice immune response in the 18 to 64-year-olds. Now compare that to the far right column, which is the black. This is a panel of human convalescent serum. And you can see, on average, that the 18 to 64-year-old titers were 3 to 4 times higher than natural infection. Again, a great sign. The 65 to 85-year-olds, they weren't quite as robust in actual number compared to the 18 to 64-year-olds. So the geometric mean titer wasn't quite as high, but it's still better than human convalescent serum. So this is, again, a really good and promising piece of information. If you look at the severe disease category, so if you look at the individuals who had severe disease, 9 of them happened in the placebo group. 1 person in the vaccine group had severe disease. So we are preventing severe disease. We're starting to get some information on other vaccines. I am at the mercy of the press for some of this data. I have not seen the official data on these early efficacy pieces of data. Moderna sounds like it's going to be very similar to Pfizer. They've published numbers, but not the raw data yet. But interestingly, now we're starting to get some information on some of the world's adenovirus vectored studies as well. Sputnik 5 is publishing 92% early efficacy. Most of you have probably heard some of the information about the Oxford AstraZeneca vaccine that's based on the chimpanzee adenovirus vaccine. They had 70% efficacy. You know, that looks low, but remember, FDA was going to be pretty happy if we had 50% or better efficacy, so it's still better than that. And they had some kind of quirky data that people are trying to understand. Just because I know you guys are going to be doing the Janssen trial, I wanted to show you a little bit of the data with Janssen. This is something that you can see. There are some preprints that are in this reference from September. And this was a study where they were looking at some of the immunogenicity in the ELISA and neutralizing titers in blood from the phase 1-2 people. And after the first dose, you can see that they had 2 doses they were studying this, 5 times 10 to the 10th or 1 times 10 to the 11th. The antibody titers in the ELISAs were actually not that much better, or actually somewhat less than human convalescent serum. And when you looked at the neutralizing titers, they were still less than the average that they were seeing in human convalescent serum. So this is after 1 dose. And Janssen initially thought that they were going to be able to get by with 1 dose. We'll see. This is preliminary data, smaller numbers of people, but I think this antibody titer is not as robust as they're seeing with other studies. And so you can see that they started with the Ensemble 1 trial, which was going to be 30,000 people that got a single dose, and they have rapidly pivoted to doing a 2-dose study with another 30,000 people. And they chose the 5 times 10 to the 10th viral particles per dose data. And I think this is where you know, knowing what's going on in the media, even when you don't see the exact data, you can kind of start to see some of the strategy. Safety and reactogenicity, for those of you that don't know the world of vaccines, reactogenicity is all of the common things that we see with vaccines: sore arm, feeling fatigued, headache, flu-like things. Safety is when we're starting to really start thinking about some of these more worrisome events like transverse myelitis or Guillain-Barré or unexpected medical events. The Pfizer EUA data has a lot that shows with reactogenicity, and I told you about the ability of these mRNA vaccines to trigger the innate immune system and that cytokine cascade. I think the reactogenicity kind of confirms that. And you can see these vaccines are going to be ones that are associated with some of the sore arm. I think 78% or something had sore arm. But, 15%, 16% had fever, 60% had fatigue, 52% had headache. These are a little more reactogenic than the flu vaccine. It's not out of the realm of what we have seen with other approved vaccines. For example, the Shingrix vaccine is one that can be associated with these more significant side effects. I can tell you just from personal experience, watching these people, the timing is pretty obvious. I mean, it starts within hours of the vaccine, the sore arm in particular. Fevers tend to really occur within the first 24 hours and then they dissipate pretty quickly. My subjects will tell me that the fever and that body ache and flu-like feeling just turned off. So it's something to expect, and it's something we'll have to educate the public about. One of the things you can see is these vaccines, the first dose is a little bit easier to tolerate than the second dose, so that'll be important education for the public as well. What I don't have in this slide is the younger populations actually had more reactogenicity than older. The older people tolerated them just a little bit better. I don't want to say tolerated, they experienced fewer side effects, but even the young people tolerated this pretty well. Janssen's data from their phase 1/2 study, they too did see systemic events like the headache, fatigue, fever. In their cohorts, this is the young people, they had about 11% that had severe grade 3 reactions. So I think you can expect that with your study as well. Some of the more significant safety data that we've seen, Pfizer did point out in the EUA document that there were 4 cases of Bell's palsy in the Pfizer vaccinated group. There were none in the placebo group, but when you actually looked at the statistical significance of that, this is actually pretty comparable to what you see in the general population. But it is interesting that the cases all occurred in the vaccine group, and so this is kind of something that FDA will be paying specific attention to as the vaccine rolls out. AstraZeneca, we all heard about the 2 cases that put the studies on hold, both in Britain and the US. Unfortunately, I think they had more information leaked than they wanted. 1 case had transverse myelitis. 1 was reported to be a case of multiple sclerosis. Some people were a little more worried that maybe, was it really MS? Could it be something more like transverse myelitis or another neurologic syndrome? Both the EU and US did decide to allow the trials to continue. Janssen, they got put on hold for 1 SAE. They've been a little bit quieter about what that event was. You guys may know that better than I. You may be also under non-disclosure agreements, which you should be. So just something, we are watching this. The trials are a really nice way of getting very, very characterized data sets. And the other thing that's really important to know is we have 30 to 40,000 people in each of these studies. So these are big studies, and we're going to be able to see important events. It won't be perfect. It's not going to always show you the 1 in a million, but it will show you a lot of safety data. We still have a lot of safety data to learn, or a lot of data left to learn. Obviously, we would like to have more long-term safety data. We are going to be continuing to follow the study participants for 2 years. FDA and CDC have worked to develop post-marketing safety systems to capture some of those rare events that will come out when you have much larger swaths of the population vaccinated. We are absolutely going to need to understand the durability of the antibody responses. We don't have data on that. We're going to have to understand the efficacy in reducing viral transmission. We think that will come about, but we don't know that for sure. We're going to have to understand safety of repeat dosing in a year if that turns out to be needed because of a decline in antibody titers. And obviously, eventually, we're going to have problems where there's going to be supply chain issues and people might get vaccinated with one company's vaccine. And what happens when you have to finish with another? Those are studies that we'll do as we move forward, but it's something that will be inevitable because of all of these supply chain issues that we have with vaccines every day, even today before COVID-19. And then obviously we're going to have to look at safety in children, pregnant women, and special populations. For those of you that are maths geniuses, there are some really nice modeling studies that are really important pieces of information that ACIP and others are using to help guide how to get these vaccines distributed in the best way we can into the population. They are going to be scarce resources for quite a while. And this is a nice way that they've shown that one of the things we were lucky is if you look at these graphs, because the vaccine efficacies are so good, it means we're going to start to be able to damp down symptomatic infections or deaths with fewer people vaccinated in the US population. You know, for symptomatic infections, even getting into 40% of the population is going to help. We want more, and that will help us really squelch the whole COVID epidemic. But these severe events will be damped down much more quickly because these vaccines are as effective as they are. There are many other vaccines that are in later stages of development. I'm not going to talk about these different ones. Actually, today, we got some of the information on efficacy for Sinovac, which is just the Chinese took SARS-CoV-2, the virus, and they dumped some formalin in it to inactivate it. It's got a preliminary efficacy of 86% according to the New York Times. So others may have better data. But I think the really important thing is down at the bottom is we need a variety of vaccines, because even when you look at the priority populations of who to vaccinate, healthcare workers, those 65 and older, those with chronic conditions, those that are essential workers, maintaining the food supply and teaching our kids, it's over 180 million people. That's more than half of the US population. So we need doses to be coming. And so I think the strategy of investing in multiple different vaccines was a good one. And that's where I'm going to stop. And so thank you for your attention, and I wish you all the best of luck in recruiting for your Janssen trial.
Thank you, Dr. Wingauer. That was a wonderful presentation.
We're going to move on into the panel discussion. I've seen about 7 or 8 questions come across the screen. So Bob, why don't you go ahead and start moderating the questions?
So we've had a number of good questions come in, and I appreciate if you would log your questions in the chat box if you have them, and we will try to go through as many of those as we can get through in the next little bit. First, Dr. Patterson, would you talk just a moment about the vaccine trial that you're getting ready to launch here at UAMS?
We are about to embark on the Janssen clinical trial, which is labeled the Ensemble. It's targeting 40,000 individuals across the globe, and it's using the AD26 vector. So it will be a single injection, and then participants will be followed for the next 2 years. You know, it's the study is thus far been very quick to enroll, and so we're really excited to be able to participate in this, and we're looking forward to being able to enroll participants that are of diverse populations, and particularly elderly diverse, and elderly meaning above the age of 60. So and that can be people who have had COVID before, or have not had COVID. That is not one of the criteria that that's not one of the exclusion criteria. So so I'm happy to answer any more specific questions about that.
All right, thank you. It's you know, exciting to see this additional vaccines get into the trial, the phase 3 trial stage. As Dr. Winkauer has pointed out, I think we can't reemphasize enough, we're going to need a lot of vaccines, and likely from a number of different directions if we're going to get the COVID vaccines or COVID situation under control. So as Dr. Winkauer reviewed, these vaccines all help our cells to produce antigen that then stimulates our immune systems to respond. Either Dr. Winkauer or Dr. Patterson, would you comment? The question was raised, as our cells produce those antigens and they're released and our immune system you know, then responds to those spike protein antigens, do we also see some immune response potentially on the cells that produce those spike proteins? Or do we know?
You know, so this is one of those issues where it's not, you know, they're presented in conjunction with your MHC. And so typically you don't create an immune response to your own cells. There could be some, but that's not a bad thing, because again, what it is, the body is recognizing the antigen. I see that the other question they worried about is this why you're developing autoimmune reactions? And all vaccines have the potential of creating some of these autoimmune disorders. It's a little puzzling exactly what it is. Sometimes it is that the antigen that is being expressed, the spike protein, mimics something in the human body. And that's something we've seen with some other vaccines. Sometimes it's theoretical. Sometimes they are able to dice it out in the lab to prove that. Sometimes it's really not clear exactly what part of the vaccine is stimulating that autoimmune response. And I would say that's the more common scenario, where you don't ever really find a specific reason. You know, maybe it's a contaminant in that vaccine, because a lot of these vaccines, not these in particular, but others, are made in cells. We're always trying to figure that out.
So we've, you know, we're basing our efficacy on these vaccines on vaccinating a number of patients and looking at development of disease. Is this really enough information to look at vaccine efficacy, or do we need to be thinking about some sort of a challenge trial to confirm this efficacy?
You know, I think we've had pretty good challenge going on in the United States. We certainly have had some pretty good challenges in Iowa. So,
right.
I don't think we need to. Early on, people wanted to do that because they thought it would speed the production of vaccine and you wouldn't have to go through these months of follow-up, but nature took its course and it did our own natural challenge.
So I'd like to turn a question in a slightly different direction, and I want to ask you, Dr. Gittens. You know, we've seen an increased incidence of death and disability in people of color due to COVID-19. The trials have been actively trying to recruit a representative population. How can we as the healthcare community support vaccination and support getting our populations of people of color involved and protected from this COVID disease without helping them to, or making them feel marginalized?
Absolutely. Thank you for Dr. Hopkins for allowing me the opportunity to be on this panel and to discuss this issue. As you're aware, the COVID pandemic has simply illuminated and or exacerbated healthcare disparities. They've always been there, but when you have incidents like this, it really does bring it to the forefront with regard to the disparity with regard to infections and significant loss of life at a disproportionate level. And unfortunately, it's going to impact us in a couple of ways with regard to vaccine production and the trials, because it's going to be a challenge for us to trust, when I say us, I'm talking about persons of color, to trust academic medicine or the healthcare systems with regard to participating in trials. And on the back end as well, once there is a vaccine that's approved and it's being distributed, it's also going to be a challenge to get people to take the vaccine as well. But so, you know, particularly the issue is the problem. With regard to how do we do that, it's I don't have a silver bullet, if you will, but I think that we have to leverage the relationships that we're continuing to build within our communities. We have to think about where persons of color, where we're talking about Latinx or African Americans, where they get their information, where do they trust, and that's thinking about our interfaith and our church base and our faith-based groups and things like that is getting out into those communities. But I think it's looking at social media and all those other counter messages that are out there. How do we combat those with more constructive messaging? But more importantly, we have to recognize that we can't be episodic in this work. We have to continuously, and I think that we've done a good job here with TRI, especially with the CABs and things like that, to continuously to build these trusting relationships so that we're better positioned for, you know, COVID-20 or COVID-21, okay, in the future. So I think that's the way. We have to figure out where they get their information from, who they trust, and find and partner with leaders and organizations and nonprofits and the like in those spaces to get the messaging to those communities.
Open conversations and longitudinal conversations.
Absolutely.
You know, there's no one shot, so to speak, vaccine to fix these issues.
Right.
So a couple of questions that have come in that I think are important for us to talk about a moment. You know, we've talked about these messenger RNA vaccines being included in lipid nanoparticles. Would there be any concern about patients on lipid-lowering therapies affecting the efficacy of these vaccines in a lipid nanoparticle?
Christine, do you want to take that one?
I'm actually going to defer to you, Pat.
That will not be a problem. These vaccines are taken up into the cells really quickly. And your lipid lowering drugs are really affecting your liver and production of lipids and stuff. That will not be a problem. One of the good things. So take your lipid drugs.
Take your lipid drugs, absolutely. Prevention at all sources.
Mm-hmm.
So there are a couple of comments about, you know, the confidence intervals in the data that's been released so far is pretty wide, particularly on our older adults. Clearly, there's a smaller sample size in some of those populations. Is this, do you think, a sample size effect, or do you think that there are other factors that play into that?
No, I think the confidence intervals is entirely the sample size, because, you know, when you were getting into the 75 and older population, they only had 5 cases of symptomatic disease. They're probably doing what they're supposed to be doing, which is socially distancing and wearing masks and washing their hands. So you will see that confidence interval improve as you acquire more data. I was actually impressed though, when you looked at the data, they had 10,000 people that they were following that were 65 and older. So it was not a teeny study. It's just, you're looking at a rare event.
Yeah, I think that the fact that at least what we're seeing here in our area is the elderly are the people who really are following the guidelines. They are wearing their masks. They are socially distancing. They are staying home. And so the number of events are going to be smaller in that group. Therefore, the confidence intervals are going to be much wider.
So here's, I think, a particularly apropos question, both for Dr. Winkower and Dr. Patterson. We have a number of patients, a number of people in our community that are research subjects for one or another of these vaccines in a blinded fashion. What would be your counsel to persons that have been part of a trial as we start to see vaccines released under EUA?
So that is the question of the day. And since we did the Pfizer vaccine trial, that question is coming up very quickly. I honestly think that it's going to be the better part of valor to unblind people that request to be unblinded and offer them active vaccine if they got placebo. I think you have an ethical issue. These people will either, many of them are in that phase 1A group where they're going to be offered vaccine in their health center or their long-term care facility, and they will just drop out. I'd rather get data in a very organized way. And so we hope, and I think what I've been told is that the VRBPAC group is going to be reviewing that and commenting on that tomorrow in an open forum that will be live streamed by FDA. So I have my fingers crossed that they're going to agree with me and we will be able to unblind. If we don't, people are going to drop out.
There was a suggestion that I had heard that rather than unblinded, just give all 30,000 participants or however many in the trial yet another series of 2 shots, but reverse the roles. The ones that had vaccine originally get placebo, and the ones that had placebo get vaccine. And that way they could continue to collect data in a different way.
Yeah, I think you can do that. I don't think it really actually does that much good, to be honest. When you think about it, they're not going to have the placebo control. That's the value of the one-to-one randomization is, if you develop cases of severe disease in the vaccine group, you're not going to be able to compare that to the control group. So I've seen that. Initially, it struck my fancy. I logistically was thinking about it, and it's like, oh boy, this is going to be an interesting challenge to add that one back on. But then I started thinking about it, and I just don't know that it adds anything. Christine, your comments?
No, I agree. Again, because you're not gonna have that control group. I think what allowing people unblinding people so that previously that got the placebo, then allowing them to get the active compound. I think what you're going to end up doing is asking different questions. And rather than your initial primary endpoint, I think that's going to be more of a short-term question that's going to be asked and answered, and then we're going to just change what our endpoints are and questions that we're asking. They're going to be more looking at the longevity and of the protective effects. So I think that the questions that the studies are going to answer are going to be different once everybody in the study are vaccinated.
I think the other thing that is an
So-
interesting one, and Bob, you might have some comments on this one because you've been in some of the meetings. One of the other options that they've talked about is that they stagger the unblinding according to your risk group and what you would receive in the normal rolling out of, I don't know whether you can call rolling out a vaccine in a pandemic normal, but rolling out the vaccine. And the only thing I'm a little worried about as the PI is that puts us in a really difficult decision, because there are going to be people that are gray, that they probably might be able to get it. And it's going to be really hard to be the bad guy and say, oh, gee, I'm sorry, you're a dentist. You're not enough of a healthcare worker, even though you're the highest risk group in the country for contracting COVID. And so I worry about just the relationships with the participants. And
Well, let me pose one final question. You've mentioned pregnant women and children being groups that we need further study on. I think we all need to think also about what about vaccinating cancer patients and others with immune compromise at this point.
so from my perspective, there are two things that you're thinking about with a vaccine. One is safety and one is efficacy. And knowing the science of the vaccines and how we vaccinate with other vaccines, these are not live virus vaccines. So the safety is probably going to be fairly similar. The efficacy is going to be harder to say. If they're on immunosuppressants, their efficacy is probably going to be less. Though in other situations, we still vaccinate with the hope that we will stimulate some immune response and it is as protective as we can get. So it's a good question. I think safety-wise, we're probably okay.
I've had very similar thoughts, and if we're talking about vaccinating 90-year-olds in nursing homes, how different is their immune response going to be in that situation too? Well, I want to thank all of you all for participating in this panel, Dr. Gittens, Dr. Patterson, Dr. Winkauer. I've learned a lot from this, and I'm sure the others have. Sorry we weren't able to get to everyone's questions. Dr. James, any closing remarks before we wind up?
No, I just appreciate everybody's attendance. We've had over 300 people participating. So Pat, you're very popular here in Arkansas. We hope to bring you back again someday, face to face. The last slide just gives a contact phone number for those that want to hear more about the Janssen study. So the study is close to launch. We'll probably be enrolling by Monday, but if you're interested in hearing more, please jot down this phone number. And thanks again, Pat.
Thank you.
Good afternoon, everyone.